হ্যাঁ হ্যালো। হ্যাঁ, আচ্ছা আমার এটা একটু প্রবলেম করছিল। আমি স্লাইডটা শেয়ার করার জন্য আমি দেখছি। আচ্ছা, আমি am I audible?
Yes, sir.
My slide is visible.
No, sir. Slide is
Hello?
not visible. Sir, slide is not visible.
But am I audible na?
Yes, sir. Yes, sir. Audible. You are audible.
Okay, just 1 minute. My sharing of slide is getting some problem. So now I am leaving and one more time I joined.
Okay, sir. Okay.
Because sharing of site is creating some problem. Now
Okay,
leaving
okay,
and 1 second I am joined.
Okay, sir. Okay.
Now my slide is visible. Hello. Hello.
Not visible yet.
Not visible yet.
Yeah.
এই এক মিনিট দেখো তো, স্লাইডটা শো করতে পারছি না, কিন্তু কি প্রবলেম ক্রিয়েট করছে। Now my slide is visible. Hello.
No, sir.
Huh?
No, sir. No, sir. Slide is not visible.
So can I send this slide to any of your email ID?
Okay, okay, sir. Okay, sir. Okay. You will send me.
Please tell your email ID.
Jyoti Bhatta 2
JU
JYOTI
JY
OTI
JYOTI
BHA
BHA
TTA 2 @gmail.com.
@gmail.com. Now I'm sending here.
Okay, sir.
But here showing is sharing your screen, but I don't know what happened actually.
Okay, sir. Some technical error, I think.
But
Please send me, sir.
Now also. Okay. I have already sent. Hello.
Sir, okay. Sir, I have not received.
But the mail has been sent to you. JYOTIBHATTA.
Sir, JYO. OTI.
JYOTIBHATTA 2@gmail.com. Already sent to you.
But I have not received till now. Okay, sir, receive, sir.
Receipt na?
Mm-mm.
Please full screen the PPD.
Okay, sir. So now it is visible.
Okay. So sorry for that particular inconvenience, because starting the session, if I have to see all the portion, then that type of problems not arises. So anyway, good morning everybody. I am heartily welcome all the participants in the faculty development program uh, uh, organized by the Faculty of Pharmaceutical Sciences, Assam Downtown University, in the same theme, paradigm shift in pharmaceutical education and research in post-pandemic era. And I am thankful to Professor Dr. Biplab Kumar De, Chairman, Dr. Minmoy Mosa, Joint Organizing Secretary, Dr. Santa Mondol, Joint Organizing Secretary, for giving me an opportunity to be here. And I also thankful to Dr. Dhrubajyoti Sarkar, my ex-colleague, for requesting me to be present here. So already 20 minutes late. Now I have to start my lecture topic. The title of the today lecture is the temperature responsive hydrogel in biomedical application. So in this title, four important words are there. That is number one, temperature; number two, responsive; number three, hydrogel; and number four is the biomedical. So first, I come to my main topic is the hydrogel. So what is hydrogel? Hydrogel means that is some carrier materials or the gel or simply the cross-linked polymeric network structure containing the water molecules. And that carrier molecules or the hydrogen, we can incorporate it, drug, or specifically any type of cell, means for delivery of the drug molecules or for tissue engineering purposes, we can deliver the drug, means a cell, through this hydrogel carrier. Next one, coming to the responsive. Responsive means sensitive, means hydrogel having different kinds of hydrogels are there. But today the particular topic is temperature responsive hydrogel, means that hydrogen or the carrier system sensitive or responsive to the particular temperature, means in response to the temperature, that hydrogen system can be swell or dissolve and releasing the drug or the cell. And that temperature responsive hydrogen system and their application, biomedical means their drug delivery, and particularly in the tissue engineering system, we have to discuss. So please next slide. Hello, please next slide. So before going to that of the temperature responsive hydrogen system, we have to know some basic portion. So what is solutions? Means polymeric solutions particularly, gels and the hydrogens. So we have to know these three things, means polymeric solution, gels and the hydrogen. So these all are containing the polymeric system. So first I come to the solutions. The left figure is the representation of uncross-linked polymeric chains. If they have good affinity with water, their meeting produces a solution. It means a liquid composed of polymer chains, each one completely surrounded by water molecules. Moreover, it can be more or less viscous as a function of the amount of the polymer that is dissolved. Simply, I giving up of the example. You know, polymer, that is the sodium alginate, Zealand gum, xanthan gum, sodium carboxymethyl cellulose, locust bean gum. So all you know the natural polymers of the polysaccharide. If we taking up of the suppose simply sodium alginate and put into the solution, then what happened? That sodium alginate having what? COOH and OH groups are there, that is hydrophilic in nature, and they are having the affinity to the water molecules, and ultimately the water molecules surrounded by that of the polymeric network and weighted and ultimately goes into the solution, and that sodium alginate making up of the solution. If the less amount of the sodium alginate is there, then what happened? The polymeric solution is the less viscous. If we increasing the concentration of the sodium alginate or Zealand gum or xanthan gum like that, then what happened? The concentration is increases and they are making up of the more viscous solution. But these are the polymeric solution. Next, coming to the gels and the hydrogen. What is gels? If polymers are cross-linked, some links between chains are established. Cross-linked, this is very, very important. If the polymeric chains are cross-linked, then the links are existed between the chains. Then gels are dilute cross-linked system, and that cross-linked structure it presents in the any solvent system, then that is making up of the gel system. And that gel having two types. One is the organogels and another is the hydrogels. Organogels means where the medium is the organic phase and where the medium is the water, means that gels containing if organic phase, that is known as the organogels, and if the gels containing the water, then that is known as the hydrogels. But simply the gels are the dilute cross-linked systems. But in cases of the, if we specifically apply to that of the hydrogens or the organogels, where there is a strong cross-linking occurs in that case. But there is a hydrogen is the strong cross-linking gels and that containing the water molecules. When solvent meets this structure, the chains are not able to separate and leave, but the affinity with the liquid left it enter more and more, causing a swelling of the three-dimensional network. Just giving up a few simple examples. Sodium alginate. If we putting into the water, making up of the part, that is a sodium alginate solution. You take one beaker and take water. Prepare sodium alginate solution. In another beaker, you take the calcium chloride or aluminum chloride multivalent cation solution. Then you put that sodium alginate solution to the calcium chloride solution or the aluminum chloride solution. Then there is a cross-linking taking place. Means that sodium alginate containing COOH group and there is a calcium Ca dipositive. So between Ca dipositive and the COOH minus, there is a cross-linking taking place. That gel is known as the calcium alginate bond. That is the cross-linked sodium alginate. This is calcium alginate. This is the hydrogel system. Means if we taking up of the calcium chloride and sodium alginate, you taking up of in the syringe, put drop by drop into the calcium chloride solution, then getting up of the spherical beads or hydrogel beads. So that hydrogen is dry up and again you put into the water. So that means taking up of the water more and more, but their structure not destroyed. So that is the hydrogel system. So that is when solvent meets the structure, the chains are not able to separate and let, but the affinity with the liquid, let it enter more and more water can be entered, causing a swelling of the three-dimensional network. So in cases of the gel and the hydrogel, that is the three-dimensional cross-linked network structure is formed. Characteristics of gels depend on polymer and on linkage density. The more cross-linked the material, the more elastic and stronger it is low swelling. So if the hydrogen that is having a most cross-linking densities are there, so that is more elastic and stronger, and the swelling ability of the hydrogel is or the gel is less because they resist to enter the water because cross-linking density is more. While low cross-linking produces very soft and weak gels, means high swelling. Next slide. Hello, next slide. So that is hydrogel I am telling. So you can think means you have everybody means we have seen the hydrogel system in our everyday life. Gummy bears we have seen that contain collagen, a super absorbent natural polymer extracted from flesh and connective tissue of mammals. Disposable napkins is based on super absorbent synthetic polymers. So that inside material of disposable napkin is a synthetic polymer called sodium polyacrylate. Polyacrylate is the means acrylate is the monomer. If we cross-link it, means that polymerization taking place, then polyacrylate form. So this is having the hydrogen, means having water absorbing capacity, means disposable napkins are the example of the hydrogen system. Sanitary napkins contain cellulose, cotton, plastic, fragrance, and antibacterial agents. The polymer powder as an additional powerful absorbent or that is super absorbent polymer that turns into gel when moistened. So all these materials you have seen, these are the hydrogen system and we see in our everyday life. Next slide. Water crystals are based on super absorbent synthetic polymer called that is polyacrylamide. We know the acrylamide is the monomer. Polyacrylamide is the polymerization or the cross-linking taking places between the acrylamide monomer and forming the polyacrylamide cross-link of structure of acrylamide polymer. So this is the water crystals. This is the hydrogel system and super absorbent, absorbing huge amount of the water. Collagen means present in the gummy bear. Cellulose present in sanitary napkins. Sodium polyacrylate present into the disposable napkin. Polyacrylamide present in water crystal are all polymers, different in terms of chain structure, number and strength of links in the cross-link structure. So all are the polymers, but varying their water absorbing capacity and solubility is different from gummy bears to sanitary napkins to disposable napkins to water cup crystal, because the polymers and their cross-linking structure or the density is different. They all are characterized by capacity of absorbing great quantity of water, but depending of their structure, the rate of absorption and the percentage of absorbed water is different. Previously, I have told because they are having different types of polymer, their cross-linking density and structure also different. That's why their water absorbing capacity and swelling capacity is also different. Accordingly, also the appearance of the gel is different. It means swollen pure collagen, that is gelatin, is very soft, while swollen water crystal are soft but less than gelatin. Next slide. And that hydrogel system are currently used in numerous applications. This is not particularly thermosensitive hydrogel. That is a particular topic coming to the later. But overall, this is hydrogel system applied in particularly different fields. They are applied in transdermal drug delivery, pH sensitive or other hydrogel system. In transdermal patch, we can apply. If we telling the thermosensitive hydrogel system, just solution form, we take up the polymer solution and the drug we have to incorporate. Suppose any wound is there in the transdermal skin, just you apply it as a liquid formulation. In the skin temperature, they convert into the gel formulation because thermosensitive, and that retain there for a long time and releasing the drug for a longer period. So there is a different types of hydrogel system that can be applied to the transdermal drug. Contact lenses, and in the polymeric, that is cross-link structure, that is hydrogel system. So that is also prepared for the contact lenses. Wound care product. So in wound dressing purposes, they are simply the wound dressing materials or the any antibacterial agent or antimicrobial agent that can be incorporated into the hydrogel system, and that is applied into the wound, because that hydrogel system is just like a tissue-like structure, soft in nature. So if you apply into the wound, there is no hemorrhage or the that is compatible with that of the wound system. So in the wound care product, there is a lot of hydrogel systems are applied. And in the tissue engineering means what actually the tissue engineering part is there. Suppose in our body, different types of tissue, bone tissue, cartilage tissue, nerve tissue, liver tissue, heart tissue, vascular tissue, so skin tissue, different types of tissues are there. So if damage of tissue taking place, so for that cases, we have to repair that type of tissue. We can apply the hydrogel system. Here what happens? That hydrogel system we can use as a scaffold material. Scaffold means the base material where the cell means that is suppose a cartilage cell we have to repair. So chondrocyte cell we can be said means that hydrogel system giving up of the environment, and ultimately where the damage is occurred, so that hydrogel system we can implant it there, and ultimately that cartilage cell or bone cell or any other cell that can be growing up. And ultimately, that hydrogel system is the polymeric system that is biodegradable in nature. And that cell, if we are growing up there, and that tissue can be repaired up, and the polymer is biodegradable and removed from the body. So ultimately, the different types of tissue can be repaired. And that is nowadays the that tissue engineering purposes, that is the hydrogel system used up in a lot. And in cases of the oral drug delivery system, that is the hydrogel based tablet, capsule, beads, nano dosage form, all are used in oral drug delivery system. And in the bioelectrode, biological means that electrode nowadays that is made out of the polymeric hydrogel system. Solar cell also, that is we are using the hydrogel based system. In the agricultural field also, that for growing up of the plant means we can giving up of the vitamin, nutrients, and all these through the hydrogel based systems like that. So these are the current uses of the application of the hydrogel based system. Next slide. So now coming to just pinpoint like that way to that of my lecture means lecture topic. So next is that is what are hydrogels? A hydrogel is a cross-linked network of hydrophilic polymers that can swell in water and hold a large amount of water while maintaining the structure. Two-line definition that is very, very important. Hydrogel is a cross-linked network. If the polymeric systems are there, that is not hydrogel. Sodium oxalate, PLGA, then xanthan gum, zilan gum, that are not hydrogel. If we cross-linked that polymer, then that polymeric system turned into hydrogen. But there should be hydrophilic polymer. Means water-loving properties should be there. Suppose sodium oxalate, zilan gum, then N-isopropyl acrylamide, then poloxoma. So if we cross-link, then what happens? That type of polymeric system containing OH, NH2, COOH, or SO3 H group. This is hydrophilic or water-loving properties are there. So that type of polymer, if we cross-link, that can swell in water and hold large amount of water. Means that can retain in large quantity of water and maintaining their three-dimensional structure. So that is the definition of the hydrogel. Water must be at least 10% of total volume or weight. Minimum 10% of water should be there. Cross-link junction points where more than two polymeric chains cross. Means cross-linking junction point you see in the figure, points are there. Means there is a more than two polymeric chains cross over. Absorbing water depends on number of the hydrophilic functional groups in the polymer bed. That is NH2, COOH, OH, CONH2, CONH, SO3. So this type of hydrophilic functional groups are present into the hydrogen system because that groups are having the hydrophilic and water loving capacity, and they can be absorb water due to their water loving capacity. Water absorption is influenced by temperature, pH, ionic state, and solvent. Means how much quantity of absorb means water can absorb, that can be influenced by temperature, pH, ionic state, and also solvent that can dictate it. Resistance to dissolution depends on cross-link density between the network chains. Means that cross-linking density means suppose sodium alginate cross-linking with the calcium chloride. So that calcium COOH, they are cross-linking. So that strength or density of this cross-linking point determine how fast or how slow the hydrogen system dissolve up into the water system or the aqueous system. Water can be free to diffuse or be loosely bound or tightly associated to the network. Next slide. Next is the advantages of hydrogen. Structural and compositional similarity to extracellular matrix. This is very, very important because that hydrogen system, that is actually the watery polymeric water system, and that is soft and tissue-like just structure. So if we apply or taking up internal or applying to supposing the nasal cavity or eye or any wound positions like that. So that just behave like that of that tissue structure of eye, wound, or the nasal or the oral cavity like that. So that is structural and compositional similarity to extracellular matrix of tissue. Extensive framework for cell proliferation and survival. So that specifically telling for tissue engineering application. Suppose the scaffold materials means that is the matrix materials giving up of the hydrogen system. And inside there, the cell is seeded. And ultimately that can be implanted where the injury taking place and that can be growing up or repair the tissue or the cell like that. So that matrix material or the scaffold material having cell or tissue-like structure and there the cell can be easily survive and proliferate. Means extensive framework for cell proliferation and survival. Next is the non-thrombogenic. Means material to generate blood clotting when in contact with the blood. Suppose that is we are applying in our biological system, that hydrogen system. So that is external materials, means enter from external to the internal position. Some materials are there. They are forming, means generate blood clotting when contact with the blood, means contact with the blood, means thrombogenic properties. But these hydrogen systems are non-thrombogenic, means not clotting the blood. Next is the biocompatible. Biocompatible is the big word, means very, very big term. Means everybody we are telling biocompatible. Biocompatible means no. Biocompatible means the in our biological system that hydrogen materials are harmony with or compatible or not. Means they are not reacting with in our biological system. Means they are compatible in our biological system. Means hydrogens are biocompatible and biodegradable. Biodegradable means they are, we are taking off internally. Means after use up, that should be removed from the body. Means they are degraded and suppose there is the polymer out there. They are converting it to monomer and then that monomer ultimately removed from the body. Means that is biodegradable. Means hydrogen systems are biodegradable. Good transport of nutrients to cell and products from cells. Means there there means that structure is the porous structure, means if that system means where the cells are treated for tissue engineering purposes, means in scaffold material. So that means nutrients and other vitamins or minerals that can be transported from cell to cell easily like that, or medium to cell easily, and can be injected in vivo as a liquid that gels at body temperature. That is our special interest, thermosensitive institute gel. Means suppose in any places injury or anything happen, so as a room temperature, that hydrogel system is a solution. Polymeric structure, drug, cell or anything is there. So we are applying as a liquid to the particular area, and that body temperature, that liquid is converting into the gel. So this is the very big point or the advantage of the hydrogel system and for the temperature sensitive hydrogel system. Next slide. Next is the types of hydrogen. So how many types are there in the hydrogen? Based on method of preparation, we can divide homopolymeric hydrogen, copolymeric hydrogen, and number 3 is the interpenetrating polymer network hydrogen. So this is the based on method of preparation. Homopolymer means same type of monomer unit, repeating and cross-link and polymerize and getting out of the hydrogen system. This is known as the homopolymeric hydrogen system. Acrylic acid, polymerize, acrylic acid. This is the homopolymeric hydrogen. Acrylamide is the monomer. Acrylamide, polyacrylamide, cross-linking, polymerize, and getting off of the hydrogen. Polyacrylamide means cross-link network of single monomer type. You see the figure in the above topmost portion in the left-hand side. Monomer is the cross-link and three-dimensional structure forming. That is the homopolymer. Next, number 2 is the copolymer. Cross-linking of 2 or more monomer units, at least one of which is hydrophilic, to render them soluble. Where what happened? 2 or more monomer units are crossed in the copolymer system and making up of the hydrogen system. And there block copolymer, alternate copolymer, random copolymer. You see the figure in right hand side top corner. So you see in the block copolymer, red, blue, and green. Prop block, 3 red, 3 blue, 3 green. Means 3 monomer type blockly cross-link and making up of the hydrogen. This is the block copolymer. Next is the alternate copolymer. Red, gray, blue, green copolymer. Then red, then blue, then green, then red, then blue, green. Means 2 or more monomer alternately copolymerize and forming the hydrogen system or three-dimensional cross-link structure. That is the alternate copolymeric hydrogen system. Third one is the random copolymer. 2 or more monomers are there, and randomly they can be cross-linked or polymerized and making up of the three-dimensional structure. Red, blue, red, blue, green, blue, red, like that. So this is the copolymeric hydrogen system. Next is the interpenetrating polymer network. What the definition told? 2 or more polymeric units in the network in which the polymers are cross-linked with each other. This is very, very important in our hydrogel-based system. Interpenetrating polymer network. What they tell? 2 or more polymeric system independently cross-linked and forming a polymeric network structure. That is known as the interpenetrating polymer network. That is full IPN or the semi-IPN. I am giving one example, then you can getting off clear. Sodium alginate and chitosan, two natural polymer we are taking and solubilized in aqueous medium and cross-linked with calcium chloride solution. Here what happens? Semi-IPN network is because in IPN formation, two or more polymeric network structure should be there. What happened here? Sodium alginate and chitosan cross-linked with calcium chloride. So what happened? Sodium alginate cross-linked with calcium chloride because sodium alginate containing COOH. COOH and C dipositive cross-linked with each other. But chitosan not containing COOH, but they are not cross-linked with the calcium ion. So one is cross-linked calcium and sodium alginate, and another is the polymer network intercalated with the calcium alginate cross-linked structure. That is known as the semi-IPN hydrogel system. Second one, full IPN. We are taking out sodium alginate and xylan gum. Both of them contain COOH group and cross-linked with the calcium chloride solution. So calcium chloride cross-linked with the sodium alginate and also calcium cross-linked with the xylan gum. So two independent cross-linked structure are intercalated. So that is known as the full interpenetrating polymer network structure. Next slide. Next is the based on environmental response. Hydrogel can be response to surrounding environment like temperature, pH, and presence of electrolyte. Which stimuli sensitive hydrogel or stimuli responsive hydrogel can display changes in their swelling behavior of the network structure according to the external environment. So what happened? Hydrogens are. So that hydrogen can be responsive or sensitive to some environmental condition. Suppose if the hydrogen system surrounded by glucose environment and that sensitive to that glucose, then that type of hydrogen system we can tell glucose sensitive or glucose responsive hydrogen system. If the hydrogen system responsive to magnetic field, then we can tell that the hydrogen system magnetic field responsive. If the hydrogen system sensitive or responsive to antibody, then the hydrogen system we can tell antibody responsive or antibody sensitive hydrogen system. If the hydrogen system responsive to metal, then that type of hydrogen we can tell metal responsive hydrogen. If the hydrogen system responsive to urea, then urea sensitive hydrogen. If the hydrogen system responsive to electrical field, then the hydrogen system is electrical field responsive hydrogen. If the hydrogen system is responsive to temperature, then the hydrogen system we can tell temperature responsive hydrogen. This is my today topic, particularly temperature responsive or temperature sensitive hydrogen system. Next, if the hydrogen system sensitive to light radiation, then that type of hydrogel we can take light radiation sensitive hydrogel system. If the hydrogel system sensitive to pH, then that type of hydrogel system which can tell pH responsive hydrogel system. And similarly, ultrasonic radiation sensitive hydrogel system, ionic strength sensitive hydrogel system, inflammation sensitive hydrogel system. So many responsive hydrogel systems are there, and research is going on in all the responsive hydrogel system throughout the world. Next slide. So, stimulus hydrogel mechanism. pH is the stimulus. That hydrogel is the acidic or basic hydrogel. Change in pH means environmental pH change, swelling, and release of the drug takes place. If the stimulus is ionic state, that is ionic hydrogen, changes in the ionic state, change in concentration of ions inside gel, change in swelling, release of drug, chemical species, hydrogel containing electron accepting groups are. So if the hydrogel surrounded environment is the electron donating groups are there. So electron donating compound means formation of charge or transfer complex taking place. Change in swelling, release of the drug can take place. Enzyme substrate, hydrogel containing immobilized enzymes. Substrate present, enzymatic conversion, product changes, swelling of gel, release of drug. Suppose stimulus is the magnetic hydrogen, that is magnetic particles disperse in alginate microscopes. Applied magnetic field, change in force in gels, change in swelling, release of the drug. Thermoresponsive hydrogen, change in temperature, change in polymer and water polymer interaction, change in swelling, release of the drug. Electrical, polyelectrolyte hydrogel, applied electrical field, membrane charging, electrophoresis of charged drug, change in swelling, release of drug. Ultrasound irradiation, that is ultrasound irradiation, temperature increase, release of the drug can take place. Next slide. And different types of tissue locations are there where the hydrogel system can be applied. Ocular, nasal, oral cavity, transdermal, subcutaneous implants, vagina, stoma, small intestine, colon, rectum. So these are the different locations of the body where the hydrogel system can be applied. Next slide. Next is the core, that is the lecture, specifically core portion, temperature responsive hydrogels. Polymers that change their solubility in dependence of the temperature are called thermoresponsive. Means if the polymer solubility can change with respect to temperature, then that type of polymer we can tell thermoresponsive polymer. The temperature at which the polymeric structure or the solubility changes is called transition temperature. Means the temperature where the solubility of the polymer can change. So that particular temperature or the point is called transition temperature. Since the solubility changes at this point is also called critical solution temperature. Means at that particular temperature, the solubility of the polymer can change, and that system can change from solution to gel or gel to solution. That's why that particular temperature is called the critical solution temperature. Generally, thermosensitive polymers contain both hydrophobic and hydrophilic components in structure, and the thermal response is the balance between the hydrophobic and hydrophilic portions of the monomer. Very, very important point. Means that thermoresponsive or the thermoresponsive polymer containing both hydrophilic portion and the lipophilic components or the portions. That response is depend upon the balance between the hydrophobic and the hydrophilic portions of the monomer. Temperature changes the interaction between hydrophilic and hydrophobic segments with water molecules, and thus can induce and change in the solubility of the cross-linked network, causing the salt to gel phase transition. What that telling means that temperature changes, means when the temperature change, that interact the hydrophilic and hydrophobic segments of the polymer molecules and the water molecules that can interact and induce the solubility of the cross-linked network, and that causing the salt to gel phase transition. Next slide. That salt phase is defined as a flowing fluid. Solution phase means what? Flowing fluid. While the gel phase is what? Flowing means non-flowing and maintain its integrity. Means salt phase is the flowing fluid, but gel phase is the non-flowing and maintain that integrity. Hydrogens exhibits separation from solution and solid either at lower critical solution temperature or at the upper critical solution temperature. What that sentence telling? That hydrogens means thermosensitive hydrogen exhibits separation from solution and solid, and that can taking places at specific temperature, that is critical temperature. Means that is taking 2 points. One is the lower critical solution temperature and another is the upper critical solution temperature. What is lower critical solution temperature and what is the upper critical solution temperature? Lower critical solution temperature means suppose one polymer solution is there at the low temperature, that is solution. If we increase the temperature at particular temperature, that converting solution to the solid gel or the hydrogen, that is known as the lower critical solution temperature, LCS. Means lower temperature to higher temperature if we increasing, converting solution to the gel. And another is the upper critical solution temperature. Some polymers are there. If we decrease the temperature, then their solution converting to the gel form. And that temperature, the conversion of solution to the gel taking place, that is known as the upper critical solution temperature. Means if the polymeric solution higher temperature is there, we are decreasing the temperature, then at certain temperature that solution converting to the gel or solid form. This temperature is known as the upper critical solution temperature. The polymer is soluble below the lower critical solution temperature, while it begins to shrink hydrophobic and insoluble when temperature above LCS, thus resulting in the formation of LCS. Previously, I have told all of you can getting up. In contrast, the hydrogen formed upon cooling of the polymer solution has an upper critical solution temperature. Means when we warming up the solution, that solution converted to the gel at particular temperature, that is known as the lower critical solution temperature. When we cool down the polymeric solution to higher temperature to lower temperature, at particular temperature, the solution is converting to the gel. That is known as the upper critical solution temperature. More specifically, the polymer in the solution near the critical temperature exhibits a phase change from a soluble state to an insoluble state. Means that conversion solution, the polymeric structure is random coil, but in the gel state, that is the collapse or micelle from they are getting off. Next slide. The hydrogel formulation injected locally that avoids surgical intervention and avoid first-pass metabolism. Means advantages of thermosensitive hydrogels, because in that cases part there is a present into the solution form. So injected we can locally where requirement is there, and their avoidance of the first pass means the drug should not goes into the liver, first pass metabolism can be avoided. The temperature induced salt gel transition is safer in the body, and it does not require any denaturing or the cross-linking, because here cross-linking is produced due to temperature changing. So there is no use of any cross-linking agent. So that is ultimately safer in the body. Encapsulation in a flowing state allow the uniform dispersion of therapeutic agent in hydrogel, because in that cases hydrogel system in the solution forms are there. So any drug or any other biological macromolecule, so that can easily or uniformly disperse into the system, and encapsulation is high in that cases. Rapid salt to gel transition at body temperature avoids initially fast release of the therapeutic agent, thereby controlling the release behavior. Means what happened after entrance into the body. So there what happened? So rapid salt to gel transition takes place, and there is a avoidance of that fast release of the drugs, and that is controlling the release of the drug can be takes place. The flowable state of the administration imparts the hydrogel safe controllability. Safe controllability means suppose in a tissue damage are taking place, or any other special tissue portions are there. Their particular shape is there, triangular, spherical, or any other shape are there. Because we are injecting as a solution form, and after injection there is a gel formation taking place. So whatever the shapes are there in the applicable spaces, so that shapes can taking up by the hydrogel system. Next slide. Thermogelling system serves as a depot once injected into the body, mitigating the need for multiple daily dosing. Sustained release of drug is possible. Means where that hydrogen system acts as a depot formation, and from there the drug is liberated for a longer period of time, and there is no requirement for the multiple daily dosings. The water content within the matrix serves as a system for drug delivery, and lubrication is sensitive area of the body, because hydrogel system we know everybody that is watery content is there, and just like tissue-like structure. So for that purposes, that is giving out the lubrication, and that is applicable to the sensitive areas or the portions of the body. Most thermogels are manufactured with the use of aqueous solvent and are physically cross-linked in the presence of heat, thereby mitigating the need of organic solvent. So in cases of the specifically the thermosensitive or temperature responsive hydrogel system, There is no cross-linking agent. Here, we can use up of heat to cross-link the hydrophobic polymeric segments, so that we can avoid the organic solvents for the manufacturing or the preparation of the hydrogen system. This facilitates the delivery of protein, peptides, and genes via thermogels by decreasing the risks of denaturations. Next slide. However, some limitations also there in thermosensitive hydrogels or the temperature responsive hydrogel systems. The use of single thermoresponsive polymers in drug delivery applications that have some drawbacks which need to be overcome. High gelation concentration, which affects both manufacturing cost and convenience of applications. This is one of the disadvantages of thermoresponsive hydrogel-based system. Poor gel strength in some polymeric systems, resulting in very low viscosity gels, which are unsuitable for certain applications, particularly single thermosensitive polymers, that giving this type of disadvantage. High gelation temperature that do not correlate with physiological body temperature, therefore not facilitating in situ gelations. So hybridization means combination of two or more polymers of these single thermoresponsive polymers has allowed for the creation of tailor-made systems with physico-mechanical properties precisely suited to the needs of the system. So for that purposes, we have to combine the two or more polymeric system to prepare thermoresponsive hydrogen that can overcome the drawbacks of single thermoresponsive polymeric system. So these in situ gel forming systems have been shown to serve as excellent matrices for cell and encapsulation, tissue engineering, targeted and sustained delivery of both drugs and genes. Their salt phase at ambient temperatures enables ease of application by injection at the desired site of action, and their ability to release drugs and genes in a sustained manner allow for less frequent dosing and administration, thereby contributing toward improved patient compliance and treatment efficacy. Next slide. Next is that classification and particularly the polymer types for preparation of thermosensitive hydrogen system. That temperature sensitive divided basically two. One is the negatively thermosensitive and another is the positively thermosensitive hydrogen system. Negatively thermosensitive hydrogen system means that hydrogen systems or the polymeric system above certain temperature means threshold that can be converting into the solution to the gel, and specifically that temperature is the lower critical solution temperature, and below this LCST or the lower critical solution temperature the polymers are soluble. Above the LCST that becomes increasingly hydrophobic and insoluble, leading to gel formation. So simply, very simple, in negatively sensitive hydrogen systems means that is LCST means below the LCST that is the solution form, and above the LCST there is the gel formation. Next slide. So that LCST or the negatively thermosensitive or the temperature sensitive hydrogen that sinks along with the temperature. If temperature increases, then what happens? That polymeric system or the structure can be shrink up. Means when a polymer dissolves in water, there are three types of interactions are there. So polymers are there. So between polymer, there is an interaction. Polymer and water, there is an interaction. And the water molecule, there is interaction there. Means if we dissolve, means that hydrogen system or polymeric system in the water. So polymer, water, and water. That type of interaction may occur. At lower temperature, what happens? In specifically negatively thermosensitive hydrogen. Hydrogen bonding between hydrophilic groups of the polymer chain and water molecules dominate at lower temperature, leading to the dissolution in the water, means that polymer soluble or dissolved into the water, because the hydrogen bonding between the polymer and the water molecules dominate, and that can be dissolved into the water. However, with increasing temperature, hydrophobic interaction among hydrophobic groups become dominated and strengthened, but hydrogen bonding become weaker, causing division. At a lower temperature, what happens? The hydrogen bonding between polymer and water stronger, that dissolve into the water. If temperature increases, hydrophobic part of the polymer, that interaction is stronger, and hydrogen bonding between polymer, hydrophilic part of the polymer and water molecule is weaker. So what happened? The association of the polymer, hydrophobic polymer chain taking places, then that converting to the random coil to the helix structure of the polymer, and that is converting into the gel. You see the structure below the LCHC, the random coil structure of polymer are there. But above the LCHC, the hydrophobic portions are attracted to each other, means that is stronger, and helix structure are formed, and that is insoluble and converting into the gel. Next slide. Negative thermosensitive polymer, that is one just here example, synthetic polymer, that is poly N isopropyl acrylamide. This is the negatively thermosensitive polymer, and that producing the hydrogen system, negative thermosensitive hydrogen, they are forming. That poly N isopropyl acrylamide, that monomer N isopropyl acrylamide containing that NaCO group, acrylamide group, and another is the acrylamide monomer, and CH3, CH2, CH3, that is propyl moieties, amide moieties and propyl moieties. Both of these are present. Amide is the hydrophilic group, and propyl group is the hydrophobic group. So poly N isopropyl acrylamide is an attractive prospect for drug delivery application due to LCSD of approximately 32 degrees centigrade, close to the physiological human body temperature. Means below this 32 degrees centigrade temperature, poly N isopropyl acrylamide soluble in the water. But above this 32 degrees centigrade temperature, that is converting into the gel or hydrogen, means temperature responsive hydrogen system, and we can apply to the different application field. Next slide. Below the LCSD, water molecules solvate the hydrophilic amide group, because that N isopropyl acrylamide group containing amide group. So below the LCSD, that hydrophilic amide group hydrated and having good water solubility, flexible, extended coil form, and hydration cell, that well structure. And in aqueous solution, hydrogen bonding between amide group and water molecule takes place. Above this temperature, that is insoluble in water and present in globular form, because that hydrophobic propyl groups interact with each other, and that is stronger, and hydrogen bond between the water and the amide weaken, and that is attracting to each other and forming the gel structure. The chains within the polymer structure collapse, and there is a loss of water from the system, and this volume phase transition can take place at approximately 32 degrees centigrade temperature. You see the figure, then you can see, clearly understood. Poly in isopropyl, amide group, isopropyl group. And below the LCST, there is a hydrogen bonding between water molecules and the amide group, and ultimately soluble. But in cases of the above temperature, hydrogen bond is weaker and water is expelled from the polymeric system, and their propyl group attracting with each other and come, and ultimately that is insoluble and converting into the gel of the hydrogen system. Next slide. The LCST of the poly N isopropyl acrylamide is closer to the body temperature and can be altered by copolymerizing with other hydrophilic and hydrophobic co-monomer. Copolymerization, what they tell you, suppose the temperature of the LCST is the poly N isopropyl acrylamide is 32 degrees Celsius, but our physiological temperature is 37 degrees centigrade. So that LCST we can change by adding up of some hydrophilic monomer to there to close to the 37 degrees centigrade temperature. Means by addition of the hydrophilic monomer increases hydrophilicity of the polymer, and stronger polymer water interaction causes increases of LCST. And the addition of more hydrophobic monomer results in lower LCST, means below 32 degrees centigrade, compared with the poly N isopropyl acrylamide. The hydrogel based on poly N isopropyl acrylamide copolymer with acrylic and polyacrylic acid one develop to adjust the LCST closer to body temperature. Next slide. Next is the another negative thermosensitive hydrogen system, means that is PPO based system. Fluoronic. Everybody we know, means we are working in the pharmaceutical field and present in our laboratory. Poloxamer, different gradings of the fluoronic polymers we are having. So fluoronic, that is commonly poloxamer. Tri-block copolymer. Tri-block means polyethylene oxide, polypropylene oxide, polyethylene oxide. This is chain, three polymeric chain are arranged together and polymerized. So consisting hydrophobic polypropylene oxide PPO at the center with hydrophilic polyethylene oxide and either side. Means polyethylene oxide two sides and polypropylene oxide is the middle side. The polymers owe their amphiphilic structure to the hydrophilic ethylene oxide and the hydrophobic propylene oxide. The gelation mechanism of PPO block copolymers could be changes in micellar properties as a function of both concentration and temperature. They can form the micelle. Means after formation of the micelle, they can be converted into the solution to the gel form and getting up of the hydrogen system. Next slide. So that amphiphilic block copolymer can self-assemble and to form the micelle in the aqueous solution. But here, for preparation of our manufacturing of the hydrogen, there is a requirement of the minimum concentration of the polymeric solution. If below this concentration is there, so if we increase the temperature also, there is no gel formation of the hydrogen formation. So here two things is required. One is the minimum concentration of the polymeric solution, and also minimum temperature required to change the solution form to the hydrogel formation, means the micelle formation. So above certain concentration, termed as the critical micelle concentration, the polymer molecules which are previously in solution aggregates and form micelle. Moreover, the micelle formation is strong temperature dependent. Below certain temperature, termed as the critical micelle temperature, both ethylene and propylene oxide blocks are hydrated, and propylene oxide is relatively soluble in water. With temperature increases, polypropylene oxide chains become less soluble, resulting is the micelle formations. Just you see the figure and clearly understood what happened there. You monomer is there, that is poloxamer. Center point is what? Propylene oxide block. Two sides you see, that is ethylene oxide is there. And they are, when temperature is low, they are present into the solution, and they are hydrogen bonding between the water and polymer taking place. But you see, when temperature increases, their micelle formation start, means above certain critical solution temperature. That center point you see, there is the propylene oxide block, that is hydrophobic. They are attached together in the center point, and externally that is extended the ethylene oxide block, means that is hydrophilic portion, and that micelle taking place. If we further increase the temperature, then what happens? That micelle coming close together, and ultimately the gel formation can taking place and converting to the hydrogel formation. Next slide. Hello, next slide. Next is the positive thermosensitive hydrogen. Previously, I discussed the negative thermosensitive hydrogen. Next is the positive thermosensitive hydrogen. Positive, just you remember, negative means we are heating the polymeric solution from lower temperature to higher temperature, converting gel at certain temperature. And another, that is LCS, negative thermosensitive hydrogen. If we cool down the hotted polymeric solution from higher temperature to lower temperature, at particular temperature, that polymeric solution converted into gel. That is known as the upper critical solution temperature. So that type of hydrogel-based system is known as positive thermosensitive hydrogel. So positive thermosensitive hydrogel possesses the upper critical solution temperature. You see the figure. Above the means temperature, the polymeric solutions of the hydrogel system in solution form. Decreasing temperature below this temperature, UCSP, that is self-assembled, and their gel formation can be taken place. Various naturally occurring polymers such as gelatin, agarose, and amylose would present a salt to gel transition as the ambient temperature decreases. Polymers possessing an UCST behavior are insoluble in water below this temperature and become soluble around. So many times I have told, below UCST in aqueous solution, it requires strong supramolecular interactions, van der Waals forces, pi-pi stacking, hydrogen bonding, and electrostatic interactions are there, means below UCST, and which are weakened upon heating, which leads to phase transition. Next slide. So gelatin, one of the example of positive thermosensitive polymers. Gelatin possessing thermoreversible properties. When the temperature is increased above 40 degrees centigrade, the conformation ranges from a helix to random flexible coil, presenting liquid. Means when heating the gelatin solution above 40 degrees centigrade temperature, that is transforming from random flexible coil to helix structure to random flexible coil. When the temperature decreased below UCST 35 degrees centigrade, some segments of the gelatin molecule are respinned into a helix, means self-assembled, and hydrogen bonds are formed between the helical segment to form a stable collagen supercoil structure, thus forming a three-dimensional networks. Next slide. Next is the preparation of the thermosensitive hydrogens. How we prepare the thermosensitive hydrogens. So many preparation methods are there. I am not coming to the details because if we go to the literature and the internet and the journal paper, you getting of the law. But here I am just giving out of all of you just brief idea. What is that system? How prepare and how we apply it in our research field like that. So that means thermosensitive hydrogen system we can prepare by heating or cooling a polymer solution. Physically cross-linked gels are formed when cooling hot solution of gelatin or carrageenan. Simply, we have to cooling the hot solution, then getting out of the hydrogen. The gel formation is due to helix formation, associate of the helix and forming junction zone due to the hydrophobic interaction and hydrophobic molecules. When groups are coming to close to each other and converting from salt to gel. Carrageenan in hot solution above the melting transition temperature is present as random coil conformation. Upon cooling, it transforms to rigid helical rods. In presence of salt, K plus, Na plus, double helix further aggregates to form C. So, if when we cool down the solution, the hydrogen system can be formed. And another, if we add the K plus or the Na plus ion, so their stronger hydrogen system can be obtained or we can prepare. Next slide. In some cases, hydrogel can also be obtained by simply warming the polymer solution. Previously, by cooling up of the polymer solution, we can prepare the hydrogen system. In some cases, just warming up of the solution, we can prepare the thermosensitive hydrogen system. Hydrogen based on poly and isopropyl acrylamide are probably the most extensive studied temperature sensitive hydrogen system. Polymers of poly N isopropyl acrylamide contain a hydrophilic amide and a hydrophobic isopropyl group. Two groups are there. One is hydrophilic group, previously I have told, amide group, and another is the hydrophobic isopropyl group. So below the LCSP at 32 degrees centigrade temperature, below. So there is a hydrogen bonding interaction between water and amide group, and that is solid. But above the LCSP, there is a means weakening of the hydrogen bond taking place between water and amide group of the polymer. But there is a isopropyl group of the polymer. That group means associated strongly, and that hydrophobic attraction or association is stronger. And ultimately, the water is expelled from the polymeric system and converted into the gel or the hydrogen system. And in this way, the thermosensitive hydrogen systems are prepared. You see the figure at less than LCSP. Sol means transparent hydrophobic portions are there, and there hydrogen bonding interaction between hydrophilic polymer and water. But temperature increases, you see in the center there is a hydrophobic polymeric portions are aggregated due to their attraction and ultimately converting into the gel structure. Next, above the LCSP, the hydrogen bond of the cross-link network breaks. The poly and isopropyl acrylamide hydrogel discharge the water and shrink rapidly, resulting in the changes from a flowable liquid to a gel-like solid. The LCST of poly and isopropyl acrylamide is closer to the body temperature and can be altered by copolymerization. Previously, I told if we add the hydrophilic monomer, then that LCST can be increased. If we add the hydrophobic portion to that of the isopropyl polymeric polyacrylamide, then LCST of the system can be decreased. So the addition of hydrophilic monomers increases hydrophilicity of the polymer, and the stronger polymer water interaction causes the increase of LCST. The addition of more hydrophobic monomer results in a lower LCST compared with the polymer polyanisotropic acrylamide. Next slide. Soiling of hydrogel. How and we determine simply. Suppose hydrogel systems are there, three-dimensional. If we put into the water or aqueous environment, they can absorb into the water. So how we determine that how much quantity of the water or the biological fluid they can be taking off of the absorb. Simply we take the hydrogel, put into the beaker containing the water or the biological fluid. Previously you take the weight of the hydrogel system. Suppose 5 grams is there. After 30 minutes you take that hydrogel system that absorb water and blotted with the tissue paper and weight. Suppose we getting above the weight 6 grams. Then how much absorb? 6 minus 5, then 1. So percentage of soiling is hard. 1 divided by initial weight is 6 into 100. So in this way, we can calculate the soiling of the any hydrogen system. So one or more highly electronegative atoms, which results in charge asymmetry, favoring hydrogen bonding with water. Because of their hydrophilic nature, dry materials absorb water. By definition, water must constitute at least 10% of the total weight or volume for a material to be hydrogen. When the content of water exceeds 95% of the total weight, the hydrogen is said to be superabsorbent. So degree of soiling, previously I just told, you can calculate degree of soiling, weight minus dry weight divided by dry weight into 100%. Next slide. Next, just I giving a few examples of thermosensitive applications of thermosensitive hydrogel in drug delivery and tissue engineering applications. Numerous active pharmaceutical ingredients have been found over the past decades. However, many drugs exhibit low bioavailability and severe systemic toxicity after administration. In situ thermosensitive hydrogels have unique advantages over traditional drug delivery systems such as anti-tumor, transdermal, nasal, buccal, and ocular drug delivery due to high drug concentration in the required site, sustained drug release, and minimal invasive. So thermosensitive hydrogels can control the release of drug, facilitate the local drug penetration, and realize on-demand drug delivery, which have best been used at reservoir for delivery. Next slide. For treating various anterior segment eye disease, topical administration is the most preferential and accessible route attributed to its safe and convenient features. Unfortunately, conventional eye drops suffer from extremely low bioavailability by virtue inherent their eye, for instances, blinking, lacrimation, and nasolacrimal damage. Because in eye preparation, maximum cases we have to use eye drops. But in cases of the eye drops application, their bioavailability is very less than 5%. Because due to the teardiness, the maximum amount of the drug can be removed from the eye surfaces, blinking, lacrimation, and nasolacrimal. But in cases of the thermosensitive in situ hydrogel, can not only be instilled accurately and easily, like eye drops, but also prolong retention and sustained drug release, resulting in enhanced drug bioavailability. What happened? Here, you applying as a solution form. So that accurately drug can be instilled. After installation into the eye, so they converting into the what? Solution into the gel form. So that gel can be lasting for a longer time, and they giving up of the sustained drug actions like that. So bioavailability of the drug can be enhanced. Next slide. Nowadays, that age-related macular degeneration means retina damage is one of the major causes of blindness worldwide. While the incidence of AMD dramatically increased with unmet clinical medicine need. For the treatment of AMD, and intravitreal injection of antivascular endothelial growth factor drug was the primary strategy. Means VGF we have to introduce for treating this AM. However, frequent intravitreal injection of conventional anti-VEGF solution render multiple complications and poor compliance, patient compliance. Only ophthalmic drug delivery system can sustain the release of active pharmaceutical ingredient and clinical means if we deliver this VEGF in situ thermosensitive gel, then that type of problem we can overcome. Next slide. So that for AMD treatment purposes, so drug delivery system based on agarose hydrogen incorporating gold nanoparticles was developed to treat AMD. Gold nanoparticles could undergo reversible softening because of low melting point of the chosen agar. So that agarose hydrogens we are taking out, thermosensitive, that is melted at less than 45 degrees centigrade temperature. So when we applying the light sources, that presence of the photons means that thermal energy means that under visible light exposure, gold nanoparticles present inside that agarose gel could convert photons to thermal energy. So that gold nanoparticles, when absorbs the light energy, converting photon to thermal energy and increase the temperature up to 45 degrees centigrade and generate reversible softening of the agarose hydrogen. And consequently, the release of incorporated bevacizumab was promoted. The release rate could be regulated by adjusting the concentration of agarose, gold nanoparticle, exposure type, and power of light. Very simple. Just here to treating AMD, bevacizumab drug we incorporated into the agarose thermosensitive hydrogen system. And inside there, we incorporated gold nanoparticles containing bevacizumab. When that can be applied to the eye and apply the light exposure, what happens? Light, when exposing the light, that gold nanoparticles converting photon to the thermal energy, and ultimately agarose gel is softened, and from there the drug is released, and ultimately we treat this type of disease. Next slide. Cyclosporine A loaded thermosensitive hydrogel was developed based on PLGA, PEG, PLGA copolymers for inhibiting post-operating scarring after glaucoma filtration surgery. Glaucoma is what? Intraocular pressure of the eye increases. Means what? Some channel is blocked up. So through which the fluid is not liberating from the eye. That's why the pressure of the eye increases. So some drugs are there, we have to treat up, but in some cases the drug is not work out. So in that cases, some surgery is necessary. That is known as glaucoma filtration surgery. Means to clear up the channel up or incorporate the special channel for clearing off of the fluid from interior portion of the eye to the exterior portion. But the problem arises after the glaucoma filtration surgery. So that channel where the creative, they are post-operative scar formations occur. So to treat this scar formation, that thermosensitive hydrogel-based drug delivery are developed using cyclosporine A, immunosuppressant drug is used. Glaucoma filtration surgery is the creation of a new drainage channel to filter aqueous humor from the anterior chamber to the outer surface of the sclera, resulting in the formation of a subconjunctival blade as a decrease in IOP. However, the filter can fill if scar tissue forms between the conjunctiva and the sclera at the surgical site. In vitro release results suggested that the hydrogel showed a sustained release of cyclosporin A over a period of 2 months. The hydrogel was administered to New Zealand rabbits via conjunctival injection, and no ocular tissue abnormality was detected. Moreover, the cyclosporin A loaded thermosensitive hydrogel strikingly inhibited scar formation, kept stable, low IOP, and facilitated filtering blade survival over a period of 10 years. Next slide. Traditional nasal drug delivery system, including nasal drop, so having so many problems are there. Means conventional for nasal applications. So, if we administer the drugs to the nasal cavity through thermosensitive hydrogel system, having so many advantages are there. And in that particular drug delivery system, so the drug can be delivered directly to the brain and avoiding the fast metabolism, and can improve the patient compliance also. And here also we can apply the drug as a liquid form, and after application in the nasal cavity, converting into the gel, and from there the drug can be releases and releasing and reaches into the different parts or location of the body. Next slide. So one thermosensitive hydrogel based drug delivery system, berberine hydrochloride, that is antidepressant drug and hydrophobic drug, and that is developed for thermosensitive drug delivery system. So due to its hydrophobic nature, so that is complexed with the hydroxypropyl beta sulfate to improve the solubility. And after complexation, that is incorporated into the Poloxar thermosensitive hydrogel. And that hydrogel based drug delivery system showed 110-fold better bioavailability in comparison to the oral administration of the same dosage forms of the drug delivery system. And that better antidepressant activity in the rat models also showing better antidepressant activity. Next slide. And buccal administration also, different, suppose gingivitis or dental flake, so many diseases are there. We are applying the drug to the locally to the buccal cavity. So here also to avoid fast metabolism, so many advantages are there. I am not going to the deeper cases. So for thermosensitive in situ gel also here applicable and advantage here, because that solution forms we are applying and there any cavity is there. So that can be save that hydrogel that having the cavity saves are there. And they from there, the drug can be released for a longer period of time. So next slide. An in situ thermosensitive hydrogel based on Poloxamar 407 was fabricated to incorporate chlorhexidine digluconate for periodontitis treatment. After injection, the in situ gel presented an immediate salt to gel transition and filled the periodontal pocket perfectly. In vitro results indicated that the buccal drug delivery system could control the release of chlorhexidine over 10 days. In another word, trimethylchitosan beta glycerol phosphate in situ thermosensitive hydrogel to incorporate erythropoietin to treat oral mucositis. They found that the blend with 10% GP presented a slow gelation at 36 degrees centigrade, while the blends containing 20% GP underwent immediate phase transition at the same temperature. Furthermore, the hydrogel composed of 20% GP and 5% TNC, the optimal mucoadhesion, control EPO release, and in vivo wound healing features. Next slide. Next, I just intratumoral injection also means for anti-cancer drug delivery. So deliver drugs directly into the tumor and decrease the diffusion of drugs into non-target organs. Intratumoral injection using a needle could render constant drug leakage throughout the body, owing to the high density and pressure to the tumor matrix, resulting in systemic side effects. In situ thermosensitive hydrogel have been broadly investigated for intratumoral injection to overcome these shortcomings, attributed to adequate local drug concentration in the tumor site, sustained drug release, and minimal systemic side effects. Next. So here for anti-tumoral means anti-cancer drug delivery, taking the F127 hydrogel thermosensitive hydrogel system and incorporating the drug paclitaxel. And here just simply paclitaxel molecules incorporated and another paclitaxel nanocrystals and another paclitaxel microcrystal. So paclitaxel is the anti-cancer drug incorporating into the F127 thermosensitive hydrogel, just incorporating paclitaxel molecules, paclitaxel nanocrystal, and paclitaxel macromolecules. So they studied and used for the breast cancer treatment purpose. But this exhibited and they showed the results of that paclitaxel nanocrystals incorporated thermosensitive drug delivery systems gives the better drug activity here. Next slide. And last is one example giving here for tissue engineering purposes. Just here, vascular endothelial growth factor means we are giving up. Means here we have developed injectable temperature sensitive hydrogel using star cells, poly means PLGA, poly L-lactic co-glycolic acid and poly that beta methoxy polyethylene glycol copolymer thermosensitive gel developed for the vascular endothelial growth factor neovascularization. What happened? Just they take that polymeric solution, incorporate the VEGF growth factor, and they are incorporate the vascular endothelial cell and directly injected into the injured site, and there the solutions of the injected gel converted into the gel formation, staying longer there, and cell can be grow up, and from there VEG also liberated, and bone regeneration can be taking place. So in this way, the tissue engineering purposes, the thermosensitive hydrogel can be applied. So in the literature and the work, so many works are there, and the research work also going on. If you go to the literature, and you can find out the different types of research work, and you can work in your places also. Next slide. So some marketed products also here giving the thermosensitive hydrogel marketed products also there. So that is available in that just 3 or 4 we are giving. I am giving here, and so many marketed formulations also available. You can find out in the internet or the Google. Next slide. So conclusions. Research in the area of thermoresponsive polymers for drug delivery and tissue engineering have been well established in the past years. Obviously, a lot of research means occur and also going on. More recently, hydrogel exhibiting thermosensitive sol gel behavior have been reported as cell carriers for tissue engineering. Typically, aqueous solutions of hydrogel used in biomedical applications are liquid at ambient temperature and gel at 37 degrees centigrade. Many hydrogen-based drug delivery devices have been designed, studied, and in some cases even patented. However, not many have reached the market. So many work is going on, but not all the research work till now reached into the market. Huge amount of research work is also there to we can carry out and ultimately we can that can reach the we can reach means that ultimately reached into the product. A research continues for the development of hydrogen-based drug delivery system. We will definitely see significant advance in the coming decades in the both healthcare and industry that arises directly from hydrogens and tangibly improve the world. So, thank you, and thank you once again for, and you can proceed fast because some recent publications on my related to hydrogen-based drug delivery systems are there. Next, and I have published this is to related to hydrogen-based system, and next, this is ongoing work also in the Elsevier. One book project also going on that is published in next year 2023 January, that is polysaccharide hydrogens derived from animal and microbial sources for drug delivery and regenerative medicine. Next. Thank you. Thank you. Thank you once again for your
Thank you very much, sir, for the magnificent presentation and enlightening us with your knowledge and experiences. Now we can move on the question and answer session. Sir, can we take the first question?
Has please tell.
If anyone has any questions, ask him. You can turn on the microphone or the message in the chat box. If anyone has any questions, ask him.
Please, any question?
No question. Thank you very much, sir. Our faculty and faculty members. For all participants, sir, one question.
Please tell.
What will be the optimum pH of in situ formulation for ocular route?
Yes. So for ocular drug delivery system, the IPH approximately 7.2 to 7.4. Okay. So when developing the thermosensitive hydrogen system, the solution system pH within this range. Hello.
Yes, sir.
This is the pH means range is 7.2 to 7.4 because this is should be compatible to the eye because pH is at that range.
Thank you. Thank you. Thank you so much, sir.
Any question, please ask. Don't hesitate.
Sir, one question from Susan Paul. Can you use Chitosan for that reason?
Chitosan for which?
Sir, formulation for ocular route. In situ formulation for ocular route.
Very nice question. Because in that cases, when if we solubilize the chitosan, that solid means that is not soluble into the basic region. The soluble that's soluble in the acidic region, but that chitosan itself not giving up of the thermosensitive hydrogen system. When we adding up of the beta glycerophosphate means copolymerase, at that time they are showing or giving up of the thermosensitive nature. But when adding up of this beta glycerophosphate, the pH of the solution changes to the pH 7.3 to 7.4. Getting up?
Thank you, sir. Any question?
Any question? Please ask. If you not ask the question, I think I am not giving up of you. Okay, the time is also because in starting time also I am that is delayed for my reason 10 to 15 minutes. So again I am sorry for that. So
It's okay, sir.
okay, can I leave?
All participants feedback form will share in chat box. Can they fill up the form before leaving? Thank you everyone
So
for being.
now I am leaving.
Yes, sir. Thank you. Stay safe and have a nice day. Thank you.